Hello students, welcome back, and today we are going to continue our lesson on Form 5 Science Chapter 1. So previous video we learned about how to categorize the microorganism in like either it's algae, it's a virus, it's a bacteria or protozoa. And in this period we are going to look at the factor that affect the growth of the microorganism. What are the factors that affect the growth of microorganism? So of course we know that in all human or all living things we need certain temperature. I think also it's similar to human. I mean like an animal. If you are too cold also or too hot, everything will just die. So the temperature must be optimal. The humidity, the light intensity, pH value, and maybe there are some others that we haven't really know or we need to know. So these are the factors that affect the growth of microorganism. Either the microorganism growth is fast or the microorganism is slow or zero growth. So let's look at the humidity. Humidity means the water in the air. For example, in near the toilet got humid, so more bacteria can grow in the toilet or in the towel that is more humid. That's why you are always advised to dry your feet and your hand, because if you always sweat, you wear the sweat clothing, or sometimes you wear shoe, you didn't wear stocking, and then you sweat. So when you sweat, there is a humidity and water at the shoe, so eventually it will cause cooties. So you got all the skin disease because it's unclean, so the bacteria will grow. So humidity is very important. Light, of course, it depends on which some microorganisms they need light, some like dark, some like light. Okay, some got light cannot grow, some got no light it cannot grow. Where the temperature usually optimal is 35 to 40, usually for all living thing and microorganism. The temperature for 35 degrees Celsius is one of the good temperature, because it's also our temperature 37, so the bacteria grow inside us, because outside they cannot grow. pH value probably 7. If too acidic or too alkaline, the bacteria and the microorganism cannot grow. And the last one also is nutrient. The microorganism must have something to eat. If there are more things to eat, then of course the growth is more. If there are less things to eat, then there are less growth. For of course there's nutrient. More nutrient, more growth. Less nutrient, less growth. So from at here we can see there we can do 5 experiments. 1 experiment, 2 experiment, 3 experiment, 4 experiment, and 5 experiment, that we can test the factor that affect the growth of microorganism. So let's say you are a scientist and you want to say that humidity cause the growth. So maybe you want to do your hypothesis. Okay, hypothesis. You say that if the air is humid, more microorganism or the growth of microorganism will be faster. For example, let's say you plan the experiment. This is the hypothesis. Then the MV, manipulate variable, responding variable, control variable. So manipulate variable will be the presence of humidity. Okay, presence of humidity. Responding variable, the growth of bacteria, and constant variable, maybe the light or the type of bacteria. Then you draw the picture, you do the procedure 1 2 3 4. Do one put in the toilet and another one put in the maybe in the living hall. For example, the bread with the bacteria, you put one is in the near the toilet. Then after that you get the data. Then of course when you get the data, we put A and B So maybe there is T for toilet, D is for dining room. So of course the bacteria grow more near the toilet because it is more humid. So the conclusion will be that we have proof that if the air is more humid, the growth of the microorganism will be faster. So this is how we do experiment, because we are teaching science, so everything we need to do experiment, because we are not just talking, we need to prove it, we need to justify, we need to explain. That is why in the science they always ask you to explain. And then, so of course we got this very simple. I already explained the experiment. So we got experiment 1, experiment 2, experiment 3, experiment 4, experiment 5. You need to understand the concept, then it will be easier to digest the information. If you say you just memorize like this, then you will very difficult to do in your exam. You must understand how to get the hypothesis, you must understand how to get the variable, how to get the picture, how to get the procedure, how to get the data, and then the conclusion just follow back the hypothesis 1. So this is yeah. So experiment 1, 2, 3, 4. So let's look at the example that let's say this is one of the example. Most important, okay, let's do the hypothesis. Bacillus need nutrient. So of course the variable is one got nutrient, one don't have. Responding will be the growth or the number of colony. Same lah, huh? Number of colony increase means the growth increase, up to you. So you can put in 2 answers, okay? Either number of colony or the growth bacteria, or the size of the growth, or the mass of the growth, the volume of the growth. Many answers, as long as it is it can be count and it can be shown. Then we have the constant one. Probably we want to say we want to use the same microorganism. If you use different microorganism, then what you want to test, you are testing different types of microorganism. You are not testing the presence of nutrients. So of course we need constant variable. Material, blah blah blah, you just say the material. In the exam will they ask, yeah, sometimes they will ask too, so you just need to give some example. Okay, to make the story short, you just put one A, one B. So one is A, he put, one is B. Maybe A is sterilized, B is no nutrient. Okay, so A got nutrient, B got no nutrient. So when you do the experiment, what is this? Sterilize the wire loop in the Bunsen. Okay, he sterilized it so that let's say the K-bar question asks you why you want to sterilize it. To kill the bacteria. Sterilize the wire loop by heating it over. Okay, so you want to kill the bacteria first, because you don't want it to grow other thing, because sometimes it's dirty, so other thing will grow, other bacteria will grow. So we just want to get the bacillus. Okay, we don't want other things. So it's right. Let's say the question asks, so it's not mentioned in your textbook. The exam can ask you many questions. The exam can ask you what is the safety precaution. So you just say wash your hand, wear glove, sterilize, so that you can get rid of certain bacteria. These are the things that you need to do. So again, I don't know what exam will come out. Most important is you understand the concept. After that, remove the environment, allow it to cool down. Okay, right? If the question asks you why you want to let it cool down, of course you let it cool down. If the thing is not cooled down and you put in the microorganism, then the microorganism bacillus will die, then your experiment will fail. So we don't want this to happen. After you sterilize, only insert the wire loop into the bacillus. Okay, so now, okay, ready, sterilized already, it's clean, it got no extra other bacteria. So you put the bacillus in. After put in the bacillus, you put into the culture solution. So culture solution 1. After that, repeat it with step B with one is with nutrient and with non-nutrient. Okay, so this one got nutrient, This one got non-nutrient. So of course we know that A will grow more, okay, because it got nutrient, it got food, more grow. B usually it won't grow or grow very slow. So we do this and inverted the petri dish at the dark room. Okay, so this is the bacteria. So okay, so after 3 days observe it and write down the data. So of course you haven't do the experiment, so they just leave it empty. If you need to write essay, you also leave it empty, no need to fill in. But you can fill in. I won't give you wrong, usually teacher won't give you wrong, but but in the exam they ask you to write essay, you no need to fill in the data. Why? Because you haven't do the experiment. So you haven't do, you are just planning. So usually the question asks you if the person going to do how. So you just plan on it. You don't know the data because we are just planning. We haven't do it yet. So let's say we do already. Let's say lah, for example, presence of nutrient, let's say bacteria colony, I put 7. This one maybe I put 2. Okay. Is the hypothesis accepted? Yes, the hypothesis is accepted. Follow back this one. Okay. Bacillus need nutrient for it to grow. So that is the conclusion of the experiment. So this one I need to put 0. Because they say that bacillus need nutrient. That because the question didn't say bacillus need nutrient to grow faster, because I put 2. So since you say bacillus didn't grow, so I just put 0 in the experiment. Okay. Okay, so then we look to the next experiment. Okay, so this one is humidity. It is the same, so I don't want to talk again the same thing. It actually is the same thing. You get what I mean or not? Because if not, I'll be reading the whole thing again. Okay, you just put 1A, 1B, 1 near the toilet, 1 is in the dining room. But of course, you don't write in the exam near toilet. Okay, you just say put in a place that is dim and dark. For example, it's humid. For example, near the longkang or near the drain, something like that. And it's the same experiment. So I hope I want to, if you are physically with me, I will ask you to go to the whiteboard and tell me how you want to do this experiment. But this is a YouTube, so I don't know how to ask you to do this, but you can try. Maybe I can ask you to try. Okay, so I no need to repeat the thing lah. I just, it's the same thing actually. That's why if you don't understand, you will memorize. Student will start to memorize it. When you memorize it, you cannot, because this book got 285 pages. So you cannot even memorize 200 pages, but you can understand this. So it's the same thing. One is high humidity. One is low humidity. Okay. So probably high is, maybe I put here 7 lah. Why I always put 7 lah? Put 14 lah. Okay. So here maybe I put 14. Here I put 3, for example. Effect of growth. The same thing. So absence of light, presence of light. Depend on what bacteria you are in, then you write down. And then temperature. Of course, if hot, then there is no growth of microorganism. Okay, again the same thing. So maybe you want to put 5, you want to put 70, up to you. Okay, you want to put 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, up to you. So I need to do 10 experiments if I want to do it properly. I will do 10 experiments because I want to see which temperature the bacteria can grow more. That is the hypothesis. Because hypothesis here stated that the growth highest at room temperature. Okay, so that's why he just do 2 only. Room temperature is around, what is the room temperature? Around, here didn't write one room temperature. I think around 25, is it? You can Google it. I think 25, 26 or 27, 28. means your room temperature. Okay, of course it's not 16 lah. 16 means you need to on the aircon, because Malaysian people like to turn the aircon to 16. So that is, of course, there is room temperature when there are aircon. Okay, when science say room temperature is without aircon. Okay, without aircon will be around 20 plus lah, 20, 25 maybe. Any temperature, any, or you can Google the temperature. Okay, continue on. Then we have the pH value. Again, it is the same experiment that you can do the experiment, the same thing lah. But one place got high pH value, one place got low pH value, one have a neutral pH value if you want. So maybe you want to do depend on, up to you lah. Maybe I do pH value 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. So probably I just do all this experiment, then I see whether it grow, or you just want to do 7. Up to you, up to the hypothesis. Okay, grow rapid at 7. So maybe you just want to put 7. Okay, this one we put acid, this one we put alkaline, this one we put 7, pH 7, and then you can get the result. Then you write, maybe here you put is 8 colony, this one maybe is 2, this one maybe 3 colony. Okay, so again, I'm not going to repeat all this, but you need to understand, you need to explain, you need to able to tell me this. If I ask you, you must able to tell me this. Manipulated, responding, constant. You must able to tell me this. You must able to tell me this. You must able to tell me this. Now, I mean the procedure is a lot. You no need to memorize it. Okay, you just need to use your own word. You need to tell me this. You need to tell me this. Not tell me, because I scared the exam ask. So I don't know whether the exam will ask you the hypothetical variable or so on. So you need to understand it. Okay. Okay. Let's do a few questions before we continue. Okay, correct. Very good. Okay, what are microorganisms? So of course, we just usually this question is 2 mark. Okay, so it's a small or minute organism, or very small. If you write very small, so I will give correct. Okay, minute organism that cannot be seen with naked eye. Okay, so you cannot see with your naked eye. Of course, you can see with your eye if you use microscope, but if you naked means you don't use microscope, so you cannot see the thing. Name 5 group of microorganisms. Fungi, bacteria,
normal flora,
virus. Very good. Okay, so of course there are 5, so just list it out. Okay, fungi, algae, protozoa, bacteria, and virus. All of this is very small, but they just categorize it under the microorganism. There are 5 types of category. What are the characteristics used to classify it? Okay, how they classify it? Probably they look at the size. Okay, what are the characteristics? Okay, number 2B. So for example, they look at the size, the shape, and the method of reproduction. Okay, so of course there are certain category. Of course, sometimes it overlap, like binary fission. Sometimes not only virus but bacteria also got. But bacteria is bigger, virus is smaller, and virus is more towards changing the person DNA as well. So there are sometimes overlap, sometimes it's a clear cut. So the biology person will try to categorize it so that we can study. For example, like maybe like a whale. Whale is a mammal, but we don't call it as a fish, though it is like a fish, but it's a mammal. Or maybe platypus lay egg, for example, if I'm not mistaken, is it? So that even though it is a mammal, but it lay egg, for example. So there are a few of these special thing, okay? So but of course that doesn't mean we cannot categorize it, okay? We still try our best to categorize it based on the priority, and so that we can learn in the book. Name a group of microorganisms that reproduce asexually. Okay, fungi and fungi. And one more is bacteria. Bacteria and so on. State 2 structure that make up a virus. Okay, so it will be deoxyribonucleic acid, okay, which is also our DNA. Another one maybe we can say is RNA, okay, ribonucleic acid, and there is two things that the basic structure, so it's very small like our DNA. State 5 factor that affect the growth of microorganism. So the 5 factor that we already study is at, where is it? What is the 5 factor? Humidity, light, temperature, pH value, and what is one more? Nutrient. Very good. Okay, one more is nutrient. Let's go down back again. Figure 1 show two types of microorganism. This is obviously amoeba. What is the group of microorganism above? Okay, so obviously this is a protozoa. What is the structure level X? So what is X? So X is not a cell membrane. You can pause the video and try. No lah. Okay, PS is pseudopodium. This is the like the hand. I know it's like cell membrane, but it actually cell membrane, so it's correct. Okay, but he say what is X so X is pseudopodium. What is the function for it to move or engulf? Let me check. See, got the I thought last time we did before. So let's look at the amoeba for a while. Where is my amoeba? Oh, it's here. So you can look at the notes. Why here don't have one? So funny one. Here this one. Pseudopia is the hand. So this one is also pseudopia. Here, up here, okay? Because it got pseudopia, okay, also known force feed, okay, for it to move. Let's say 2 mark, okay, for it to move 1 mark, and for it to engulf during zygositosis, means for it to eat. Okay, the hand will come out, the pseudopia, and then it will put inside it. Okay, so yeah, of course there are video, but I cannot put it here because it's other people video. So you can try and check the video. So we can see in this video, for example. So let's continue the video. So of course in this video we can see that this is the what is this? Amoeba eat the paramecia, so the hand coming out already. Of course this process is very slow, so it takes a lot of time, but I think they fast forward it already. So therefore the phagocytosis, it engulf it already, so this thing cannot come out already. So let's see whether how it digests it. Okay, that's quite slow, so it got no place to move, getting smaller and smaller, and this one no place to move already, so now he start to entering and engulf the food and so on. So that will be the video for the amoeba. Okay, so that is the thing that we can see. Okay, where was I? We do until this question. Where's the question? Let's go back to the question. Here. Okay, so that will be the structure of the thing. Okay. Okay, so if no question, we are going to take a short break and I see you by maybe 5:40, okay?
Okay.
Okay, see you then. Bye bye.